Hello, my name is Dr. Lane Schreiber. I'm a physician and electrical engineer. This video, Molecular Virus Killers, is the second in a series. The first of the series was titled Take the Fight to COVID-19. In this particular video, we explored the analysis and we explore the potential designing of a molecule which could seek out, hunt down, and kill coronavirus. Coronavirus, or COVID-19, is an RNA genome. The coronavirus's genome is approximately 29,900 nucleotides in length. The nucleotides are bits of information which are strung together to generate or represent the genetic code of the virus. RNAs are made up of 4 different nucleotides. These 4 nucleotides are A, C, G, and U, which stand for adenine, cytosine, guanine, and uracil. The DNA is comprised of 4 nucleotides also. These 4 nucleotides are A, C, G, and T for adenine, cytosine, guanine, and thymine. The molecule that we're discussing in this video is transcription factor 3A, which is outlined here. Transcription factor 3A has a 5' end and a 3' end. Transcription factor 3A is made up of 365 amino acids which are strung together. Transcription factor 3A has 9 different zinc fingers. Zinc finger number 1, 2, 3, 4, and 5 are meant to bind to the DNA over the course of about 30 different nucleotides in the DNA. It binds to a particular sequence of DNA, and once that is accomplished, then zinc fingers 6, 7, 8, and 9 bind to other transcription factors. So this particular molecule will locate a segment in the DNA. Once it does that, then other transcription factors will come and build up on top of it. And then from there is constructed a much larger molecule, oftentimes made up of as many as 73 transcription factors, which is then called a transcription complex. And that transcription complex is used to decode the DNA, that is, read the genetic information that is found in the DNA, and create RNAs. RNAs then move to the cytoplasm, and in the cytoplasm they are used as templates in order to construct proteins. The COVID-19's genome is again 29,900 nucleotides in length. It is used to create proteins also. It is used to create its own shell, which is called a virion, the receptor on the surface of the shell, as well as reproduce and copy its own genome in order to make daughter viruses, which then go on and infect other cells, which then cause the infection to persist. Transcription factor 3A's job, again, is to locate 30 nucleotides in the DNA. It can be modified in order to create, in this particular case, a molecular virus killer, where we would change the coding of these 5 zinc fingers in order to hunt down, seek out a specific segment that the COVID-19's genome has, which is particular to the virus. We want to avoid interfering with RNAs that the cell normally creates in order to make its proteins. In order to do that, the key is finding amongst the 20 amino acids, and they are listed here as far as the letters that are used. We're not going to go over the names because of the short time, but the idea is to find which of these amino acids would bind most tightly to the 4 nucleotides that are used by the COVID-19's genome for its genetic information. If we can accomplish that, then we can create a transcription factor 3A molecule which will seek out, bind to, and basically arrest the replication process of COVID-19. My father, Dr. Lane Schreiber, was able to use spatial analysis and binding analysis. And what I mean by that is that in the cell, in the nucleus. The DNA is a double-stranded helix. In space, it is twisted. So therefore then, when you have amino acids that bind to the DNA, you have to account for this spatial relationship that amino acids have to nucleotides. What he was able to determine was that the nucleotide asparagine, which is represented by N, attaches to adenine, which is one of the nucleotides. That glutamic acid, which is represented by E, binds to cytosine. That arginine, which is represented by R, binds tightly to guanine. And lysine, which is represented by K, binds to thymine. So therefore then, with this code, this key, we are able to determine which of the amino acids we could use to place into the 5 zinc fingers to look for, bind to a specific string or signature of COVID-19's genome in order to have this molecule bind to COVID-19's genome. The 365 amino acids of this particular polypeptide or molecule would therefore then prevent COVID-19 from being able to replicate. If it can't replicate, the infectious process has been ground to a halt. Now, what we need is the help of engineers, of computer programmers, of biochemists and bioscientists, and the general public. Anyone who has an interest in using their analytical powers and resources to be able to determine what is the most optimum sequence of nucleotides in the COVID-19's RNA genome that would be the target to use for a molecule like this. For more information, please go to taskforcecovid19.com. This is a wicked virus. Please, my friends, be careful, be safe. Thank you very much.